Welcome to Professor SNAB, where we aim to make A-level biology as simple as a cell. So today we are looking at knowing the difference between monosaccharides, disaccharides, and polysaccharides, including glycogen and starch, being able to relate the structures of monosaccharides, disaccharides, and polysaccharides to their roles in providing and storing energy, and knowing how monosaccharides join to form disaccharides and polysaccharides through condensation reactions, forming glycosidic bonds, and how these can be split through hydrolysis reactions. This sounds like a lot, but I think this is a topic that comes together in a nice way and just makes sense once you understand it. So let's begin. So looking closer at carbohydrates. Well, basically this entire topic is about carbohydrates. So all molecules in this group contain carbon, hydrogen, and oxygen. Carbon atoms can form covalent bonds, making them stable as covalent bonds require a lot of energy to break. Carbon compounds can form small, single subunits or monomers that bond with many repeating subunits to form large molecules or polymers. So, what is polymerization? It is the process of single subunits coming together to form longer chains. This is something you probably would have come across in GCSE chemistry or even A-level chemistry if you're doing it this year. Yeah. So the three types of carbohydrates are monosaccharides, disaccharides, and polysaccharides. So what are the types of carbohydrates? We just looked at them. Monosaccharides are the monomers of carbohydrates. So a good example is glucose. The word monosaccharides in itself contains the word mono, which means one. So that is your clue there. Disaccharides, once again, the word di means two. Disaccharides are the product of two monosaccharides joining together via condensation reactions. In a condensation reaction, one water molecule is released. A glycosidic bond is the chemical bond that forms between two monosaccharides. So basically, a disaccharide has a glycosidic bond between them. To put things into perspective. And yeah, moving on. Polysaccharides are polymers, which are basically long chains of carbon. They are joined together by glycosidic bonds in condensation reaction. And examples would be starch, glycogen, and cellulose. So now, focusing on starch. Starch is a storage polysaccharide of plants. It is stored as granules in a structure known as amyloplasts. So that is all you need to know if, that point is all you need to know if you're doing AS level biology, but if you're doing A level biology, then you will come across amyloplasts once again in topic 5. Moving on, starch is constructed from two different polysaccharides, amylose and amylopectin, which of course everyone should know. So now another example of polysaccharide that we should look into is glycogen. Glycogen is a storage polysaccharide of animals and fungi. So if you have already started looking at past paper questions or practice questions, you might have come across a lot of these difference between starch and glycogen questions and or maybe features of glycogen kind of questions. So for those kind of questions, that's your first point. It is talk about how glycogen is a storage polysaccharide for animals and fungi compared to starch, which is a storage polysaccharide for plants only. Glycogen is highly branched and not coiled. It is compact, so lots of it can be stored in a small space. Again, this is something I come across so many times in mark schemes, so get used to using these sentences in your answer. So now looking closer at glucose. Glucose can come in two forms, so alpha and beta. They are almost identical except for the fact that they differ in the location of the H and the OH groups attached to carbon 1. So feel free to pause the video now and just look out for the difference, because you'll remember it more if you look at the difference yourself instead of me just telling it. So yeah, what you will notice is that on carbon 1, the OH and the H groups are interchanged um, in beta glucose compared to alpha glucose. And another top tip I would like to add quickly is that get used to practice drawing the alpha glucose molecule whenever you sit down to revise topic 1 A-level biology, because it is something that comes up in past paper questions so many times, and you don't want to miss out on 2 marks just like that. So yeah, get used to drawing alpha glucose and beta glucose, because sometimes they can ask you the molecule to draw the molecule beta glucose, and you should know the answer. So the main function is to store energy within the bonds. When the bonds are broken during respiration, energy is released. So the bonds they are talking about in this case is the covalent bonds. Glucose is soluble, so can be transported easily. It has many covalent bonds which store energy. Some monosaccharides are used to form long structural fibers, which can be used as cellular support in some cell types. So now, glycosidic bond. So this is the bond that holds 2 or more glucose molecules or any monosaccharide together. 2 hydroxyl groups on different monosaccharides interact to form a strong covalent bond, giving rise to a glycosidic bond. If the OH groups are located on carbon 1 of one monosaccharide and carbon 4 of the other, a 1,4 glycosidic bond forms. If the OH groups are located on carbon 1 of one monosaccharide and carbon 6 of the other, a 1,6 glycosidic bond forms. The glycosidic bond is broken when water is added in a hydrolysis reaction. Disaccharides. So you guys are probably familiar with what disaccharides are now. The main function of disaccharides is to provide the body with a quick release source of energy. Maltose. So you have 3 types of disaccharides that you should be familiar with. So we have maltose, which contains 2 molecules of glucose linked by a 1,4 glycosidic bond. And then you have sucrose, which contains a molecule of glucose and a molecule of fructose linked in a 1,2 glycosidic bond. And finally, you have lactose, which contains a molecule of glucose and a molecule of galactose linked in a 1,4 glycosidic bond. Easily soluble in water due to presence of lots of hydroxyl groups. This is something you probably would have come across in A-level chemistry. So yeah, this is one of those places where you will see a connection between the 2 courses. Once again, this slide is very important because you will come across countless practice questions, past paper questions, and exam questions asking you 2 markers on on disaccharides. Like there could be questions like, what is maltose made up of? Like, what are the 2 molecules in maltose? What are the 2 molecules in sucrose? What are the 2 molecules in lactose? Or they could ask you the other way around as well. So like, what 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 disaccharide do you get from 2 glucose molecules? What disaccharide do you get from a glucose molecule and a fructose molecule? What disaccharide do you get from a glucose molecule and a galactose molecule? So important slide, lots of past paper questions. Remember it. So now focusing on polysaccharides. So polysaccharides can be branched or unbranched, straight or coiled. So being branched increases the rate at which a polysaccharide can be broken down. Being straight means the molecules makes the molecules suitable for structural functions, and being coiled makes it more compact and suitable for storage function. So that's kind of the different distinctions you have between these words, and it's a good way to visualize it to make your understanding of the topic easier. So now focusing on starch and glycogen, like I probably said, I think I've said this earlier, but so many questions talking about the difference between starch and glycogen. So again, start this star this slide, highlight the slide, something. So starch and glycogen are useful as storage polysaccharides because they are compact, which means large quantities can be stored, and they are insoluble, both of them. They will have no osmotic effect, unlike glucose, which would increase the solute concentration of a cell and causing water to move in by osmosis. It doesn't do that. So now focusing on starch alone. So now we're going into the differences between starch and glycogen. So starch is stored as granules in plastids and is made from two different polysaccharides. So amylose is when unbranched helix-shaped chain with 1,4 glycosidic bonds between alpha glucose molecules. The helix shape enables it to be more compact and thus more can be stored, which is what you want. And then you have amylopectin, which is kind of the other side. A branched molecule containing 1,4 glycosidic bonds between alpha glucose molecules and 1,6 glycosidic bonds, and it has a branch structure, hence very easily hydrolyzed. Once again, a very, very important slide because you will see countless exam questions on these. So you should understand this slide a lot. So now glycogen, the storage polysaccharide of animals and fungi, and it is highly branched and not coiled. It contains both 1,4 and 1,6 glycosidic bonds. Glycogen is more branched than amylopectin, therefore more molecules can be added or removed by hydrolysis. So that is all from Professor Snap today. If you have any questions, please drop them in the comments below. I do understand this topic is slightly more difficult to wrap your head around compared to the topics you've done so far. So if you have any questions or any points you need clarification on, you have the comments. And feel free to share this video to someone who might find this useful, and don't forget to subscribe. Thank you.